So every year, millions of diabetic patients survive because of single biological hack. We took the human gene for insulin, cut it out, pasted inside the microscopic bacterium, and forced that bacteria to mass produce a human protein. This isn't science fiction, it is a recombinant DNA. And if you are studying for your university exams or any competitive test, mastering this multi-step workflow is guarantee way to secure maximum marks. So students, welcome to BioMinds, your ultimate guide to mastering molecular biology and building a high growth career in the life sciences. If you want to crack your biotech papers with ease, make sure to hit that subscribe button right now. Let's break down the architecture of recombinant DNA. So basically, to understand the rDNA technology, forget the complex textbook definitions for a moment. Think of DNA as a massive movie film strip. If you don't like a specific scene, or if you want to add a brand new scene for a completely different movie, what do? You use scissors to cut the unwanted part out, you use tape to paste a new scene in, and then you put it into projector to run the edited film. So in the genetic engineering, our scissors are restriction endonucleases, our tape is DNA ligases, and our project is a host cell like E. coli. So let's look into the genetic engineering. Okay, so I have break down this genetic engineering into multiple uh, sessions because it's a huge topic. So first today we will start with the enzymes of genetic engineering. So there are certain enzymes that we use for the genetic engineering. So see, basically genetic engineering is a cut paste technology. So what you are cutting and what you are pasting, so basically it's a DNA. So genetic engineering means you are creating something. Engineering means to create, right? And genetic means is a genes. So you are creating a genes. Okay, so you are modifying genes, you are adding, you are deleting genes so that you can get the character which you want. Okay, just like the insulin. Okay, so there are two enzymes that we use, mainly two enzymes. There are so many different enzymes we use, but main and the very common enzymes that we used in the genetic engineering are restriction endonuclease and the ligase enzyme. Rest all enzymes we will study in the another session. So this enzymes are very much important. So first enzyme is your restriction endonuclease. So it is known as the molecular Caesar, as I have already mentioned. So molecular Caesar means it actually cutting down the DNA at the molecular level. So molecules means a nucleotide, right? A small molecule. So it actually breaking down one of the bond between the molecules. So therefore it's known as the molecular Caesar. Okay, so it cut down the certain short sequences within the specified or unspecified distance. Okay, basically it's a cutting tool. Okay, it is cutting down the DNA at the specific location. See, we have a molecular Caesar here. So we have a different restriction enzymes. More than 600 restriction enzymes have been identified. Okay, so these enzymes have, so see, we have a 600 enzymes. So studying the 600 enzymes individually, it becomes very tedious. So we have classified those 600 enzymes into different categories. Okay, so we have a type 1 restriction endonuclease, we have type 2 restriction endonuclease, and we have a type 3 restriction endonuclease. So basically, this is the summarized form of the restriction types of restriction endonuclease. So here I have categorized into four categories. So I have class distinguished, I differentiate this type 1, type 2, type 3 by this four categories. Okay, by the recognition side, discovery, requirement, and the structure. So basically, type 1 restriction enzyme, see, recognition site means what? It is a specific site where that restriction endonuclease go and recognize it and cut it there. Okay, so what do you mean by recognition site? Okay, so it's a recognition site means it's a specific sequence in the DNA. Okay, So we every nucleotides, sorry, every restriction endonuclease have the specific restriction site or a recognition site, you can say. So when we have the recognition site category, so type 1 will cleave at random site. Means if you have a DNA, it will cut DNA at any point. So you will get any fragment. Sometimes you will cut down the main gene as well. Okay. Whereas a type 2 and type 3, it's just these two are a specific. Okay. So this, they cleavage, they cleave at very specific site. Okay. So means if they have a certain sequence, so that restriction and type 2 restriction or type 3 restriction endonuclease go there, identify that sequence and cut it there only. Okay. So the discovery, if you see, type 1 was discovered by Michelson and Yuan. Type 2 was discovered by Hamilton O. Smith and Associates. Next comes the requirements. As this is the enzymes, you all know enzymes require something, right? Cofactors or energy source. So like type 1 and type 3 requires three important things: ATP, magnesium ion, and S-adenosine methionine. Whereas type 2 require only Mg2 plus. Okay, so if you are working with a type 2, you have to give the Mg2 plus to it, and your reaction will occur very smoothly. And next is the structure. Type 2 is a small monomeric structure. Type 2 has the two subunits, whereas type 3 has the, it is a quite a large with the three subunits. So this is the overall differentiation of the type restriction endonuclease. So the question comes, which restriction and which type of restriction endonuclease we will use for our recombinant DNA technology or genetic engineering? Correct. This is the question where that arises. So type 2. Why type 2? There's something, there must be a reason why we are using type 2. It's because it is a specific, it will cut at the specific sequence. So you will get the gene, okay, at the very specific sequence. And it only requires Mg2 plus. It does not require ATP and S-adenosine methionine. So if you add a Mg2 plus, your reaction will occur very smoothly. Okay, so type 2 restriction endonuclease we use for rDNA technology. So this type 2 restriction endonuclease, we obtain it from the various bacterial species. Okay, so based on the which bacteria we are using, we name that particular restriction endonuclease based on the bacteria. So suppose I have an example, EcoR1. So here we have abbreviation, meaning, and the description. So here E stands for Escherichia, which is the genus. CO stands for coli, which is a species. R stands for RY13, which is the strain. And I stands for, I is not I, it's a first. It is a first identified order of identification in bacteria. So the the starting letters are for the bacteria. So EcoR1 means it is discovered in E. coli, and R is the strain of the bacteria. So this is how the nomenclature has done. Next, how the DNA has been cut using molecular CSIR. So we have the palindromic sequence. Okay, palindromic sequence means what? So here you can see we have a sequence of GTATAC. So if you read it from here or if you read it from here, it's the same. So that's known as a palindromic sequence. Okay, it's a reverse complement basically. Okay, so if you read it from the both the direction, it is actually going to be same. Okay. So how it cuts? So if you see EcoR1 has the recognition site is the GAATTC. So EcoR1 just identify the sequence GAATTC. So it will cut between the G and A and G and A. So this is how it will cut down. Okay. So after cutting down, you will get this kind of overhanging ends. Okay. So that overhanging ends are known as sticky ends. Okay, so we get the two types of ends, sticky ends and the blunt end. So overhanging ends are known as the sticky ends. Where because why it is known as the sticky end? Because it is readily stick to the other sequence, which is complementary to it. Okay. We don't require much energy for that. So this is hint 3, which is Haemophilus influenza. It is derived from the Haemophilus influenza, this restriction endonuclease. Okay, so this is the sticky end. This is also sticky end. And what is blunt end? So here it is cut here in between. So you will get no overhanging. Okay, so it's require more energy to basically join. So these are the two sticky ends, and this is the blunt end. Next comes, we have a ligase enzyme. So here we see our DNA is breaked by our restriction endonuclease. So we have a glue that can actually joins our DNA. So this joins the bond between the molecules. So that bond is known as a phosphodiester bond. So restriction endonuclease cut the phosphodiester bond, whereas ligase enzyme joins the phosphodiester bond. So here you can see this is sticky ends. Okay, so it is ligated by our glue or tip, you can say, by ligase enzyme. So we have two types of ligase enzyme. So it is depend upon the source. So if it is taken from the T4 ligase, it is a fast T4 ligase. If it is taken from the E. coli, so it's a E. coli ligase. Fast T4 ligase require ATP as an energy source. E. coli ligase require NAD as an energy source. Fast T4 joins blunt ends, whereas E. coli ligase joins sticky ends. Okay, so this is all about the two main enzymes of genetic engineering. So we will study the next part and the next session. For say, tune for it. Thank you.